We're now up to our final session for today, and we are joined by immunologist Dr. Gros. So I'll start by asking Dr. Gros to introduce himself.
Well, thank you for inviting me. It's a pleasure to be here. Really important work that you've been doing this week, and I've been following it with some interest. My qualifications, I'm a professor of immunology. I've got 50 years experience of working both in animals and in humans and all aspects of infectious disease. That's viruses, bacteria, parasites, fungi, and of course my major expertise is in the immune system and how you either make a vaccine against these infectious diseases or you use the immune system to fight cancer and things like that, or you regulate the immune system to stop it from causing allergy and asthma. It's a very complicated system, very powerful. It's in every part of our body, but it actually keeps us safe from many things. And I've had that training both here in New Zealand, in the States at the NIH, and also I've worked in Switzerland for 5 years on various aspects of human immunotherapy design, of immunotherapy products. My present employment, I'm actually just retired, and I'm a deputy chair, it's an honorary position of the Malaghan Institute Trust Board. But previous to that, I was the director and CEO of the Malaghan Institute of Medical Research. I also led the VANS, Vaccine Alliance, Aotearoa New Zealand, Ohukaitu, which is really the coming together at the time of the COVID pandemic of bringing together people that can create, to literally use the Maori translation, the shield to protect people from the virus.
Excellent. Can you just, so this is the Vaccine Alliance Aotearoa New Zealand. Can you tell us a bit more about that, how it was started and who its members are? And
I think to understand how it was formed, you've got to understand, just take your time, your minds back to what was happening when we were hearing the first reports coming out of China, that there was a new virus, a virus that could be linked to something like SARS-CoV-1, which we knew had a high lethality. And we were all concerned because we had been following. We knew there was going to be a pandemic of a virus at some stage because the way the world was globalized, the way people moved around. And we know that viruses are always cooking up something new because we're a very easy target population. So when we first started to hear the news of this new virus, unknown, with very dramatic effects, it wasn't just killing old, sick, immune-suppressed people. It was killing young doctors who were treating those people. And it was very frightening. And we didn't know what could happen to that virus. So as a group of scientists around New Zealand and people involved in vaccine manufacture and academics and biochemists, we've decided to form an alliance across all sorts of organizations to see if we could mount what would be needed for New Zealand in the event the virus came to New Zealand and how we could help in that response. I'll just detail some of the major things that we presented to government, and the major things that we were tasked with doing was, one, we had to prepare a white paper on all the virus and immunology capability that was in New Zealand, in case New Zealand was left on its own, had to rely on its own resources to mount a response or a vaccine or some kind of antiviral medication, etc. We had to form an alliance with CEPI, the Coalition for Epidemic Preparedness, which is the large international body tasked with overseeing all the vaccines being built in the world against COVID, but also any adverse reactions, any kind of problems that could be occurring. We needed to link with that international body. We had to have actually authentic, proven, credible leadership, and we hired immediately a very fortunate Dr. Fran Pretty, who had experience both in Africa and the United States in bringing new vaccines into a population and all the problems associated with that. And she really helped the clinical program and led that program and providing advice to New Zealand. We also were tasked with making a vaccine in the event there was no vaccine made available to New Zealand from elsewhere, because we didn't know at the time what vaccine would work. So we had enough expertise going from discovery, design, discovery, research and preclinical studies, all the way through to vaccine manufacturers scale up with our partner South Pacific Sierra down in Timaru. So we brought that coalition together and set about creating a pipeline of opportunity there. We also felt very important that as experts in the area of immunology and vaccine design, we had a really important role to keep abreast of the international developments and also feed that back into the community through the various forms that were operating, whether it be the Samoan Capital Radio, Ministry of Health officials. It was our task to provide that expertise, and we had that expertise for the country. And also not to close down. I think it was really important at that time. Someone had to have the lights on, and the Malaghan Institute was uniquely placed that we're independent of many other places, so we could keep the lights on and do our research that was needed.
Thank you. While we're on the topic of your, I guess, qualifications from that role and qualifications to speak to the topics we're speaking about today, I'd like to understand a bit more about your processes for reviewing scientific literature in the area. Earlier witnesses today have spoken to papers that they say have not been available elsewhere and are making sure that some of this research is made available. And I just want to ask in general terms, without going into those specific papers at the moment, but do you come across papers that ostensibly raise concerns with the safety of the vaccine or other issues, and how do you make sure that you find and see that information?
It's our duty as scientists and experts in the area to read all papers and consider all possibilities. Of course, history has always proven that sometimes you miss something or get something slightly wrong. So it behooves us to actually read everything and understand everything. Now, when COVID started, there's maybe 1 or 2 or 3 papers a month coming out. Now there are literally thousands of post-vaccine studies, etc. It is an enormous undertaking to keep up with the literature. So now we use the power of AI to actually monitor some of the things going on, and that's very important. We use the power of people tasked with writing reviews and looking at all the different clinical trials. So there's ways of consolidating the massive amounts of information. Now, you refer to papers or points of view or scientific understanding that is not in the accepted or normal mainstream published literature. Social media, for instance. Believe it or not, we keep an eye on those things too, because one can never be so arrogant that one knows everything about the human body and about infectious diseases, and certainly about viruses. So we would pride ourselves that we keep the true scientist and the scientist, and we have processes for making sure that we do review and give fair credit to people who have valid points to make. But I must make one point. For those that who do get their papers rejected, or they're not considered by mainstream journal outlets, they may want to consider that maybe they have misinterpreted, or go back and argue the case. You don't just go off into your corner and say, oh, they don't understand me, and I'll just have my own alternative. The only way we can survive as a humanity is that we actually engage together and discuss things together.
And I'll take it, some of our witnesses also referred to articles and have referred the Commission to articles that are published, but I take it from that you will consider everything that's in a published journal.
I have been very interested in this proceedings, and I've gone on the internet and listened to some of the people that were mentioned today and try to follow some of the arguments that they were making just to see what valid points they might have. So very open to those sorts of things.
Thank you. Right, we're going to ask you first to talk a little bit about vaccines in general from your expertise, and I wonder. Again, today we've heard talk of traditional vaccines being different than the particular vaccine we're looking at today. But I wonder if you could just explain to us vaccines in general, a little bit of where they come from and how they work. In very simple and brief terms.
Well, that is a difficult thing, but I think that. Most people have experienced vaccines, and vaccines is 200 to 300 years old. Some say it goes back a couple of thousand years because some wise old people realised that rubbing some crust, say in the smallpox case, from cows and that sort of thing can actually protect you from the real disease of smallpox. But it's basically about 100, 120 years old, and it's a constantly evolving science. It started off with very crude approaches where once the germ theory of a disease was understood, that actually bugs in our environment or from each other can cause quite serious disease, people set about, well, how can we use the body's natural immune system to fight that disease? And so various ways to modify, kill, attenuate, which means grow them in other organisms so they actually lose their pathogenicity for humans. Can we make vaccines that way? They're the traditional ways of 100 years ago. But as we have developed biochemical techniques, genetic techniques, we've been able to take out only those parts of a virus or a bacteria that we need to make the immune response to. And that's very important to consider. Just throwing whole bugs into a person makes a very crude, actually relatively dangerous vaccine. If one remembers the old pertussis vaccine, you put into little kids, and there can be quite violent reactions. We now have a vaccine for pertussis where it only takes out the bit that actually will neutralize the bacteria or actually causes the problem. And this is the wonderful way that the sciences of biochemistry, genetics has really interfaced with vaccinology to make safer, better vaccines, certainly over the past 20 years. I can make a point. Some vaccines cannot evolve. Things like the flu vaccine, which we're currently stuck with, it's a 30-year-old technology, grown in chicken embryos of all things. And people want to stay with that? No. We want to move on. And I think there's real possibility now that we've seen such great advances with the RNA technology, we can now move to a flu vaccine with an RNA technology. And that's very exciting for the world.
Can you take us then through the development of that technology? So when COVID came along and we didn't have a vaccine and there were efforts all around to develop a vaccine, can you talk to us about that process?
I think one myth I'd like to dispel, that somehow it's a new technology. This thing is, RNA technology is at least 30 years old. It's been applied for cancer, it's been applied to curing some quite Ebola, it's been applied to doing rabies, horrible, horrific diseases, and it's actually in clinical trials right now. So it's a 30-year history of that. Second...
Can I just pause you on that one for a second? Yeah. Again, this morning we had a witness who described that particular history as being a reason for concern, i.e. that the technology had been around for so long, but in that time we didn't have safe ways of using it to this scale as being a reason to be worried about the vaccine.
That is mischievous, I dare to suggest, because that's not what was holding it up at all. What's holding it up is, in the case of Ebola, it's a rather random disease. It's very hard to ethically start just putting a vaccine in people, and you have to wait a bit for the Ebola outbreak, if you like, and testing that. You've got to get, under our current capitalist system, you have to get the drug company to see enough money available, enough people who are going to have Ebola to actually justify the hundreds of millions of dollars they're going to have to invest to make a vaccine. If people want to, if the governments want to change that and just give a company the money to do that, an Ebola vaccine would have been made ages ago.
You're saying it would have been made with this same technology, yes, exactly. Is that your view, right?
There's been a slight, I just also want to address the lipid technology. People talk about the way...
Again, sorry, just to interrupt, that this is the LNPs that we've heard people talk about?
Into the acronyms, yes. The LNPs. That is also an old technology. In fact, from the beginning of time, we've always known that if you can encapsulate proteins or bits of the bacteria or virus in little vesicles and actually make it's called an emulsion, it actually makes the most effective vaccine. And that's called adjuvants, Freund's adjuvant, complete Freund's adjuvant, and that's a very old technology. What's been a bit of the breakthrough, it is a sort of scientists tend to be guilty of getting very excited about their one contribution to discovery, but the improvement on this has been the ability to really precisely put lipid around an RNA and very quickly to protect it, because RNA is so vulnerable and so fragile, it normally disintegrates, and that's actually been the barrier to using its power for a safe and effective vaccine. The fact that we could actually basically run the lipid and the RNA together in the same stream, and then you come out with encapsulated stuff. That was it's a funny little fluidics device.
Thank you. I wonder if you could talk to us a little bit more about the process of how the mRNA vaccine works, and just talk us through that for...
Well, I'm glad I hope people are listening to this because we just have to take our bodies, and we know that our bodies are made up of cells. And we know we're made of several hundreds of millions of cells. And each one of those cells comes with its own little nucleus, which contains DNA, and the DNA contains the genes. And I'm sorry, I've got to give you a little, just brief, the DNA, the genes, they then make little messengers that go out into the cytoplasm, the outside of the, well, not the outside of the cell, the bit before the outside of the cell. That's where proteins are made, where it's a muscle protein or a membrane surface or a receptor or whatever, and that uses messenger RNA. The DNA gets changed into a code of messenger RNA. It's a messenger. It's called messenger RNA, and it instructs the machinery in the cytoplasm of the cell to go and make a protein, which we all understand. It's like milk proteins or muscle proteins. And what's been quite clever with this RNA vaccine, or the ability to really use it, is the fact that you can put in this RNA, it goes inside the cell, and it only goes into the cytoplasm and starts making protein, the protein that we want it to make, which is the viral component, in the case of COVID, that we need to make an immune response to. It doesn't make anything else that it actually confuses the immune system. Just the spike protein as an example. Everyone remembers that word, the spike protein, which is what you do need to make an immune response to neutralize the virus. So that's the power. You can actually put in just a messenger that makes the one protein that you need to make an immune response to. What happens to that? RNA is very quickly degraded. It's within minutes to an hour. And so then that's gone. The protein lasts a lot longer. It actually is on the surface of the cell in the case of the COVID-19 vaccine. And the immune system, for reasons I don't have time to go into, the immune system of each one of us as individuals sees it as foreign and immediately starts making an immune response. An antibody response, a T helper immune response, a cytotoxic T cell response. There's a lot of different types of immune responses, which we don't have time to go into. However, it is seen as foreign to that person, and then the immune system just takes over and makes all the materials and agents that will neutralize the virus, or what it thinks is a virus. And that is destroyed. And often the cell producing that protein is destroyed.
Can I pick up on that point? Because we've heard some concerns today also that by using this mechanism, this vaccine turns our cells, I think the phrase was, turns our cells into a factory that produces spike protein, being a dangerous protein, and then also turns our immune system response against our own cells as opposed to against a foreign
body. That's the power of the
body.
immune system. That's why I don't have enough time to explain to you that the immune system, of course, every day, every second of the day is looking at things coming out of your cells to see if they're self, non-self. The immune system of our bodies has been programmed to absolutely exquisitely distinguish anything that shouldn't be there versus everything that should be there. It's amazing.
You've said just a little bit earlier that the cell that produces it may then be killed.
Yeah.
Is that a problem?
That's a normal process. All cells that actually deliver to the immune system and go to the lymph node, they eventually die. They actually have a short half-life, if you like, as well. Once they leave the skin, say, or they go up to the lymph node, and then that's it. It's a really surefire way, a safety way of making sure nothing goes on for too long.
Thank you.
Can I just make another point?
Let's go for it.
Because you stopped me, actually.
Sorry,
so,
yes.
So the RNA gets degraded and gone. The protein stimulates an immune response, a normal immune response to the virus, spike protein. And then you make a whole lot of cells that then can live in your body for many months to years, waiting in case that spike protein or a virus bearing that spike protein comes back again. And the memory response, the immune response, is so devastating, you don't even feel it when you get an infection.
This speaks to some of the other concerns that we've heard, which include that either the LNPs or the spike protein do not remain at the injection site or might stay in the body for an extended period of time.
Thank you. Can I jump in there? I just know where you're going. Because I read the papers about this.
Yes, so you have come across studies that
yes.
say that. Okay, thank you.
We're very interested to know about this. unfortunately, there's been a misreading of some of the high-quality work done in this area, because the lipids are made of lipid, and the tracer chemical that was used to follow the lipid, of course, it gets broken down, but they are just lipids that can, and it's just the tracer that people are following that goes to other parts of the body, because the body recycles and reuses fats and lipids for its own purposes, but it's not any different from any other part. It's not as though it's contaminated or in any way dangerous. It's just a normal lipid, like if you eat butter, if you eat mutton fat or something like that. It just joins the party of building our body.
So I guess if I can summarise, you have seen some papers about this, but your expert opinion is that they've been misinterpreted a little in this, and it doesn't cause you alarm or concern.
Yes, and I was actually disappointed that people have used those studies to cause, and I'm sure the authors of those papers have been disappointed to see how it's been used to alarm people, because it wouldn't take much as a scientist to realise they're talking about the lifetime of the tracer, not the lifetime of the lipid nanoparticle, the LNP particle in the body. It just dissolved pretty quickly. Otherwise, the vaccine wouldn't work.
If I may, I might take you through a few of the other concerns
Yep.
that we've heard today, and through previous evidence to the Commission. One particularly strong concern was about the overall speed at which this vaccine was able to be developed relative to development of other vaccines over time. And can you speak to that and potentially the clinical trial processes?
Yeah. I'd like to speak to it in a couple of different ways. First of all, New Zealand has always been privileged. New Zealanders have always had a privileged life. I'm not going to get narky with them, but most medicines are well trialled and been through lots of people and volunteers overseas. So we get a lovely medicine always served up to us. This time, with the COVID pandemic, in real time, New Zealanders were part of that whole process. It wasn't sped up, just that we've normally been quite spoilt in the way medicines, just make that one point. Second point, yes, thank goodness all effort was to make this the fastest process, but I would make the point. What happened was a lot of resources were put in front of the scientists and drug manufacturers to make this a seamless process between phase 1, phase 2, phase 3 clinical trials. All resources to make sure that, because normally what happens is they torture the drug companies in a way to make the vaccine. Let's have a look at it for a year or two. Then we do that, the next phase, phase 2, and then we'll look at the results. And then we had the world community of scientists could not wait for the 3 to 4 years, which is traditionally done for vaccines against organisms which we already know much about. We did not know about this organism. We needed a vaccine as quick as possible. So global effort was put in to make sure that the clinical trials were done very carefully. So I think there were 60,000 people done in the phase 2 clinical trial. It was done the regulations, the manufacture of the vaccine, all the issues around the contamination that could have happened from DNA, RNA, other things creeping to the system, were all done very carefully. The scale-up manufacturing, absolutely rigorously tested. If I think I could just go through to you, what happens is there was a lot of work done initially with the COVID long before 2019 about coronavirus spike proteins because of the SARS-CoV-1, etc. People were really terrified that this thing could come back in a really mutant form and kill a lot of people. So a lot of research had already been done. A vaccine was designed, several vaccines were designed around with messenger RNA around the spike protein. I think only one involved a membrane component, and that's the one we have. That worked. All the others didn't really work. Then it's tested in cells, tested in mice under very cold controlled situation to test whether does it integrate with DNA, does it do things that we don't expect. Every part of the immune system, every part of a mammalian organism that actually could be affected by the vaccine were tested in those preclinical studies. Done very carefully before even approaching a first-in-man trial where human volunteers were then tested, and then every aspect of those human volunteers was tested to make sure before going to the next phase of several hundreds and thousands of people to make sure the vaccine was safe, make sure the vaccine did what it was supposed to do, which is to make a neutralizing antibody against the virus. Really important, very careful process. At the same time, the drug companies that were being commissioned to make the vaccine were being rigorously tested and evaluated. Could they make a small batch of vaccine? Could they make a scaled-up version? Because it's quite a different game to make a large batch of vaccine for millions of people versus making a small number of vaccines for a few mice. That's quite a different game. So there's a lot of parallel processes to make sure globally some vaccines that could work were made available for the global population.
When it was sped up to enable that to happen fast, is there any risk associated that we don't get to see potential longer-term effects?
Very good question. I think people need to understand, when you had something like the pandemic, we didn't know we had a virus that was evolving, still evolving. We didn't know what other diseases it could cause. It infects your whole body. You've got to remember that. You go through phase 1, phase 2, 3, release, start protecting people, start protecting your vulnerable people. There's a phase 4, which means when you're testing millions and millions of people, you're always looking for those very rare individuals who may have a funny immune system or very rare life history or may have co-infections, very rare co-infections. All sorts of funny things start to pop up when you vaccinate thousands or millions of people. And that's phase 4. And so there's a whole process set up. That's what, the adverse reactions registries, which now a lot of anti-vaxxers are poring over with some glee. That's what they're all about. They've been set up by the system, by the governments, by the drug companies to make sure something isn't being missed, that it couldn't be a population which could be vulnerable to the ill effects of the vaccine.
So we've heard about those systems today, and particularly concerns that they have shown thousands of adverse reactions to the Pfizer mRNA vaccine in particular.
We have to define what adverse reactions. I'm sorry, I'm just excited now. I've got
do.
to jump in there. But adverse reactions come in many different colours and phases. It could be a bruise, it could be a red spot, or it could be that you feel dizzy for a week. There's a lot of adverse reactions, and it's really critical that they all get reported, because we shouldn't be so arrogant that we can understand it could lead to something else down the track. We know that viruses, when they infect you, for example, they can have quite tragic sequelae later, 10 years later. So we're always monitoring in the future.
And is it true to say that after the rollout of the vaccine, even in New Zealand, we did learn of some of those adverse impacts, including myocarditis from the vaccine?
We don't want to go into the details of that. It's one of the tough things to deal with, and you've got facing a pandemic with an unknown virus, which obviously could kill people, young and old people. You roll it out, and sometimes people do strange things in their lives, and the vaccine may coordinate with that and cause problems, or some people may be vulnerable to the effects of stimulating the immune system at that phase of their life. And we find that young men, and there was cases of another COVID vaccine, the AstraZeneca one, where there's certain ladies or vulnerable to a sort of a blood clotting thing. But let's get real here. Very rare, billions of doses have gone out, and we're talking a few hundred actual cases. Now, I don't want to downplay the tragedy, etc. I'm just giving the reality. That is actually part of life.
I think, as you say, with some of those tragic situations, what we've heard a little bit across the time is a concern that these vaccines were delivered to otherwise healthy people that may not have otherwise faced those consequences. Yeah. Do you have any comment on that in terms of?
This is a really tough area, and I'm reluctant to get into it too much. One thing that comforts, I do think that you don't know what would have happened if they actually got the real virus. That there may have been some vulnerability to the real virus infection. Virus really does cause cardiac inflammation. The virus does cause major issues in the lungs. The virus does cause major issues for the cardiovascular system and in the brain. We've yet to see what the consequences of this COVID infection has for our bodies long term.
Just two quick follow-up questions on that. I think in the session, or a couple of sessions before you, we had some witnesses that cast doubt on the suggestion that that the virus could cause myocarditis at higher rates than the vaccine. Do you have any knowledge of studies in that area?
Well, yeah, I read everything. Well, not
Yes.
read everything. I can't say arrogant thing to say. I try and keep up with things. I think that should be debated. Let's put it this way. That's, of course, if there's a genuine concern, that should be debated and scrutinised. And, of course, all the cardiologists around, worth their salt around the world are looking at those things. And now we have some wonderful tools to work out, has that person been affected that time or that time, related to their cardiac event or not? Time will tell, and information will come in the future to see whether there's actually plausible or causal links, etc. Let's just wait.
So is it fair to say, and I think you mentioned at the beginning, that there are now thousands of post-vaccine studies being created
thousands
regularly?
of post-vaccine studies, yeah.
So I guess, is it fair to say that there's things that we may yet be still to learn about impacts of the vaccine?
Yeah, I think that's fair to say, and I think that's what Phase 4 is all about. And the drug companies sign up to it, who made the vaccine. The government's mandated that it has to happen. MedSafe Pharmac, absolutely, that data must be collected on and on and on. All vaccines are under that regime. All medicines are under that regime as we speak. I think.
So, sorry, I'll just pause you. When you said then that the government mandated it, do you mean mandate collecting of the information as opposed to, we've been talking about vaccine mandates today, but not...
medicines out there. All medicines that we are receiving, based on Panadol, anything from Panadol to the new cancer medicines, of course, they're absolutely monitored to see who they may not work for. We are all individuals after all.
And I guess, on the other hand, is it also fair to say that there might be long-term effects of COVID that we are still, the disease itself as opposed to the vaccine, that we still...
I fear the long-term effects of COVID far more than the long-term effects of the vaccine. I think that people have to remember the vaccine stimulates an immune response in your arm, your draining lymph node, that's as far as it goes. And it makes a healthy immune response that actually protects all sites of your body. The COVID infection gets inside you, it affects all the cells lining your blood vessels, of your kidneys, of your heart, of your brain. That's a dangerous virus. I heard someone say before, oh, it's just another respiratory infection. No, it's not another respiratory infection. It's a virus that goes beyond the lung and deep into our bodies. It's excreted, we've monitored it in the sewage system. That tells you it systemically infects us. And later on, you need a good vasculature. Just think about heart disease, brain disease, dementia. We don't know what this pandemic will result to in several years' time.
And we've heard some concerns that, similarly to COVID, the vaccine that allows our body to create spike proteins might also allow spike proteins to be created all through the different tissues of the body.
That's mad. I don't want to be crude or... no, that's not been shown to occur. Let me just say that. It's been scrutinised.
In the studies that you've looked at.
Can I just tell you, FDA asks for all sorts of things like that. I have been on a drug development program, and it can be quite ridiculous, some of the things they ask you to scrutinise to make sure when you have a new medicine going out, to make sure it's really safe and doesn't do something that no one hasn't thought of.
Thank you. I'd just like to ask you about one other area of concern that we've heard about before we finish up, and this is about the residual DNA, so not the mRNA itself, but as I understand it, the plasmid DNA that's involved in the creation of the virus, and that there's some of that DNA also injected into our body along with the mRNA vaccine, and concerns about the effect that might have, including whether it could be a cause of cancer.
Yeah, these are the issues for the manufacturers, and so we have a process called GMP, Good Manufacturing Practice, and anyone making a vaccine or a medicine, a lot of medicines are made in cells where there can be DNA from the cells, or it could be animal cells and things like that. They absolutely have to meet very stringent criteria that there's only a very small percentage of DNA from, say, a host cell, for instance, in a vaccine formulation. In the case of the messenger RNA vaccines, that's bacterial plasmid DNA. It's very easy to test for. It's very easy to get the level of contamination. You can make batches of RNA, and some could be heavily contaminated and some not, but you have to have good analytical systems to make sure that you don't confuse RNA from DNA. They're two different chemicals. It's actually very hard sometimes to see the difference, and I think there's been a couple of papers which have been misinterpreted or badly done, claiming there's a lot of DNA contamination in an RNA formulation. Yes, you can make a sloppy RNA formulation, but the vaccine RNA is actually stringently go through stringent things where independent groups have to test each batch of the vaccine as it's been made, as it goes through scale-up, to make sure that it fulfills all the criteria around levels of DNA contamination, levels of other kinds of contamination, before it's released, that incredible word, before it's released for public use.
And so if things do go through this system and they've met the tests, there still can be some residual DNA? Is that the situation?
You can never say never, and it depends what you define as DNA. There could be some DNA bases, very short sequence, which are completely harmless. I think what some people are worried about, and if I could just draw my old ancient history, is large pieces of DNA that could contain a whole gene. People talk about genetically modifying something and things like that. There was a case of a polio vaccine back in the 60s. People in New Zealand were dying of polio. The public was absolutely banging on the door of the government, get a polio vaccine. And so we bought, unfortunately, a cheap polio vaccine at the time. It was contaminated with SV40 DNA. And so there's a whole lot of people in New Zealand who were given the polio vaccine with SV40 DNA. Nothing has happened, and that is large chunks of DNA. But so far, in a vaccine formulation, we have no indication that it has a way of getting into the body and transforming. Just to reassure people, DNA from bacteria and viruses, it's actually recognized by a level B, level C kind of defense mechanism of our body. We can see what a bacterial DNA looks like, and we dissolve it. We have enzymes in our cells that kill off that DNA, and the same for RNA. We don't like viral RNA either.
Thank you. I guess there's just another phrase that we've heard again today that's related, but that this particular technology with its mRNA is not a vaccine, but an injection of genetic material. Do you have any comment on that?
I think that's playing with words. Measles vaccine, which is a highly effective vaccine and saves kids' lives, contains the RNA, the ability to transcribe things. I think that we have DNA viruses. I just think that's mischievous. I just don't think we should credit.
So there are other vaccines that use genetic material in what might be considered traditional vaccines.
Yes.
Okay, thank you. Just in the last few minutes, I'll turn to commissioners to see if you want to ask any further questions.
Professor, I do have a couple of questions for you. At one or two points in your evidence, you've expressed a reluctance to get into some issues. Some people might interpret that as hiding something. Can you just explain why you're reluctant to get into some issues?
Because I think because can be very emotional. You need time to really specify what the words you're using to describe RNA, for instance. RNA means a lot of things to a lot of different people. So you need to be quite scientific and be very specific and really define things down. You can't do it in a soundbite. So I'm just reluctant to get into sound bites and spitting things out, etc. like that. I think it's more about, I'm very happy to have a deep and meaningful discussion or debate, and I wish actually there could be more opportunity to debate some of these scientific things if people are interested. We do it all the time at our conferences. We argue vociferously, but open it up to the public. It'd be great.
Right. And so there's an issue about the length of time and detail that you would need to go into in order to explain some things properly.
Yeah, exactly.
It's not that you're trying to hide anything.
No, 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 no. I think that the world gets reduced to sound bites, and I'm just reluctant to start throwing darts. I can throw darts at people, but I'd like to have a proper forum where we can actually do it in a scientific, disciplined way, where you can specify, like lawyers do. Let's define what we're talking about, and then just work through how those, using those definitions, what we're talking about, the relationships between those various things. I think that's a fair thing, and I would welcome that. I think that's really important. But it's not to have slugging matches about who can outsmart who with a few cute words.
Just in relation to RNA, we're talking here in the community vaccines about mRNA. Is there any room for confusion about that term, given that we sometimes hear about mitochondrial RNA?
Oh, okay, yes.
Can we just clarify that, please?
Yeah. So messenger RNA is a very specific thing. It's a very specific kind of chemical entity. It's got certain structures and features very different from the mitochondria, or if you like, ribosomal RNA. I would actually want to mention that, which is actually almost a structural element of a cell, and where protein is made. And it's called RNA, but it's very different from messenger RNA. It's got very different behaviours. It's very long-lived, whereas messenger RNA is designed to be a messenger. And it's the danger of getting into using acronyms like LMPs, etc., that we start to debase the discussion by relying on very, very broad acronyms, because it means a lot of things to different people.
So in relation to the non-traditional vaccines, we're talking about messenger RNA?
Yes.
And in what sense is the RNA a messenger in this situation?
It takes in this situation, we've given, it's a messenger for a piece of viral protein, which is the virus uses to enter cells. And by putting it in the way of our immune system, the immune system makes neutralizing antibodies and neutralizing T cells that can actually neutralize it when the virus comes into the body to attack the body.
Just so that we try to get a clear explanation on this for people who may be attempting to follow it. The virus has a central component, and on the outside of the virus, there's a spike protein.
Yeah. Yes.
And am I right in thinking that the messenger RNA recreates the spike protein, but not the central component of the virus?
Yeah, exactly. It's a very truncated version which can only do one job, make that spike protein, which we need to make the immune response to be safe from the virus.
So when the real virus comes along and tries to enter a cell, it does so using the spike protein?
Yes.
And there's quite a technical explanation, which I'm sure you could give us, but we'll avoid that, as to how it does that. But is it your evidence that the mRNA vaccine causes the cell to recognize that spike protein and treat it as foreign?
The immune system seeing it on the surface of the recipient cell.
Right.
So you've got to imagine, you put in the LMP containing the RNA, it gets taken up by dendritic cells. I should say, I don't want to get into the detail because there is some discussion which cells it goes into. People talk just to make it simple muscle cells, but actually there's a specialized set of immune cells that sweeps our bodies called dendritic cells. They take up the messenger RNA vaccine and actually deliver it to the lymph node. And they're the ones that actually really put it on their surface and say, hey, immune system, this is what you need to be making an immune response to. This is foreign. And that's actually how it's done.
So if the spike protein is all that's introduced by the vaccine and not the virus itself,
nothing else can be made.
Then we can't contract the virus from the vaccine.
No, it's impossible.
So in terms of safety, the important thing presumably is that the spike protein doesn't harm the body.
The spike, once again, it comes down to names. The spike protein is actually not something people imagine a spike that hurts something, because I think there's been a lot of misinformation that we're full of spike protein that can then degrade our kidneys. It's actually a very tricky little molecule, and it's actually not so much like a spike. It's once again where I have a problem with scientists being too enthusiastic about their science. It's a clever little shapeshifter that actually hides from the immune system normally. That's why when you just put a whole virus in, you don't often make a very good immune response to. And it sort of unfolds itself and cozies up. It's more a cozy molecule. We much better, they called it the cozy molecule, the cozy protein. And it just cozies up and enables to increase the affinity for the virus to get inside the cell. Right. So the idea of spike circulating around our body and spiking everything up is a complete misnomer and misinformation.
Right. So given your lengthy experience in this field and your studies of the COVID-19 or the SARS-CoV-19 2 virus, what is your view now, after all of the time that's gone by, about the safety of being vaccinated?
Oh, it's going to be called our safest vaccine we've ever made. If you look at now the billions of doses given with this vaccine, and the safety profile has been fantastic. I'm sorry for those people out there who don't want to believe that, but the safety profile compared to any other vaccine is amazing. And the efficacy in protecting people from serious disease, protecting our young people from serious disease, it has just been tremendous. It's an actually, I just think it's an amazing human endeavour what's been created. Can I just actually add, I just thought I should add conflict of interest, because I see a lot of people's opinions have been demeaned because people say, oh, they work for a drug company or this and that. I should actually just point out, I'm not employed by anyone, I'm retired, I'm not working for a drug company, I'm not anyone's payroll. I volunteer work as a trustee of the Malaghan Institute of Medical Research, just to make sure, so clear, before we have, oh, he's working in the cahoots with the drug company. I'm not, I never have been.
Right. Now, we heard you talk about contamination as a result of DNA. Some people suggest that there's residual DNA in the vials that are used. What do you say about that again?
Just to go through that, the plasmid DNA is used to make the RNA, and then it's purified away. And then there's enzymes which are specific for DNA, which can be used to degrade today, to really get it down to really small levels. There are the purification columns and reverse dialysis. There's a whole lot of purification processes that people like William Rolliston down at South Pacific Sierra really spend their whole life and resources on, purifying the thing that you want, which is the RNA, away from all the other things that were used to make the RNA in the first place. And that's a very normal process. And independent groups, independent agencies are used to test each batch and each stage of the batch to make sure there's no DNA contamination.
Some groups think that the original manufacturing process, which resulted in the Pfizer virus being approved in the US, was a different process to make the scaled-up version of the vaccine. What do you say about that?
I think that is... I'd like to know what detail they're referring to in there, because, of course, when you make a big batch of something for billions or millions of people, it's different from making 100 or 300 where you do different levels of quality control. I'm not surprised. I don't understand what the concern would be. And in fact, as you learn how to make something like this, you put the regulatory authorities have to put more and more controls in as they learn more about the process. So I think that's part of the normal evolution of making a good vaccine.
Do you have any concern about the scaling up of the vaccine into large quantities than were originally tested?
I think that if we're comparing RNA vaccines versus the traditional vaccines, like look at like with the Chinese vaccine, many thousands of litres vats of virus had to be made and purified from cells versus a 40-litre batch for the RNA for the same number of people. The levels of purification you have to do for those old traditional vaccines is far worse, far more difficult, full of far more contaminants than the new. I think the RNA process is going to win out because it's so much easier to deliver cheaply. And remember, cost is a big thing. and actually safer and full of less contaminants than the traditional vaccines made the traditional ways with cell lines and infecting cells and infecting chicken embryos.
In terms of the unintended consequences of vaccination, we now know that there are some serious side effects for some people. Tell us, please, what you consider those established side effects are.
I think the cardiomyopathy is actually a very important thing for young men. That seems to be important. I think, depending with the RNA vaccines, I should ask, what are we talking about? RNA vaccines?
Yes.
Yeah, I think that's the one that's come through. I think that depending on the dosing, the vaccine formulation can stimulate in some very sensitive people, and some people have certain genetic conditions and stuff where they have aberrant immune cells. We're finding out those people, they need to be treated very carefully if they are being vaccinated. And we're finding out what those special interest groups are, as with all medicines. And I think that if you think you've got a potential for a condition like that, you talk to your doctor and you get treated specially to make sure that you don't react in a strange way. And there's all sorts of medications to help calm an overreactive immune system if you have that kind of immune system when getting any vaccine.
Do you accept that anaphylaxis can be a side effect of the mRNA vaccine?
We don't have time to go because I'm a bit of an expert on the anaphylaxis way because I specialise in Th2 immune responses, IG, etc. It's actually not great at making traditional, what we define as anaphylaxis. That's not a fair statement from the anti-vaccine lobby group. I think that there's room for understanding that the vaccine can stimulate the inflammatory part of the immune response to excessive degrees in some case and cause some really poor feelings, and people have described feeling unwell for days and months, and that I'm perfectly willing to accept. But anaphylaxis and stimulating IG immune responses to have a genuine systemic anaphylaxis due to IgA-mediated immunity, I really find difficult to believe from all the data we've seen so far.
Finally then, you said in the course of your evidence that there's a need to engage to discuss things together.
Yes.
You've heard people say that debate and discussion was shut down during the course of the pandemic. Do you accept that there was a shutting down of debate and discussion?
When you have a public health crisis, and I'm a little bit outside my train tracks here, but the government of the day had to consider all the issues, what they're dealing with. They know how the hospital system is geared up to deal with really sick people. They know the effect on the economy. They know the number of people and groups that could be vulnerable who are in old care homes. That's a political decision about all the elements that a pandemic virus coming in unknown, and that's their role to make that decision, not my role, is what I'd say.
So if debate is shut down temporarily for the greater good. Would you support that?
If they're dealing with the right evidence, yes. Yeah.
And so the crucial question is, what evidence did they have before?
I think there was sufficient evidence supplied. The vaccine manufacturers, there were well-published clinical studies of the 60,000 people that received the virus before we even started thinking about getting that vaccine. I think there's a lot of evidence out there, and I don't understand why that was ignored when we actually had some very well-done trials to show how that vaccine worked, what it delivered, and how safe it was.
Thank you very much.
Okay, thank you very much.